Thank you for being here. I'm happy that there's more than the 3 people I know in the audience. That's always exciting. I currently work for NOAA Fisheries at the Ecosystem Services Division. I'm also a PhD candidate at the Hawaii Institute of Marine Biology. So I kind of play 2 hats right now in my life. And one of the projects I've been focused on a lot is on Cryptofauna and the diversity of these Cryptofauna. So what is biodiversity? Most people think they know exactly what it is. It's just a variety of organisms. And yeah, to an extent, that's right. It's the variety within and among life habitat or an ecosystem or a landscape. It's defined by a specified measurement that you use to understand what the diversity is within. Now, it has 2 main components, richness and evenness. To explain richness, it's just essentially what are the unique species within different environments. So here on the left, just at your right, you will see a school of manini. That's one species of fish. And then on the right-hand side is another habitat, but there are 7 fish. Therefore, the habitat on the right with the 7 fish would be more diverse than the left. Another way to explain diversity is a concept called evenness. Now here you have A and B. Within A and B, there are 4 species within each one of these plots. But as you'll observe in plot B, you've got a domination of a deciduous tree. Therefore, one would consider that A is more diverse than B because the organisms are more evenly spread. Diversity is used to describe variation in several forms, like the genetics, the species, the genes. We just talked about the species. Life forms, so as an example, in a coral reef, you can look at the diversity of different types of corals, whether they're branch-shaped. You can also look at functional groups. Not just to give you an example of what a functional richness would be. That same image on the left of the manini, that's just one type of herbivore. Whereas on the right, you've got 5 different types of herbivores. So the habitat on the right would be more functionally diverse than the one on the left. Now, diversity is expressed in several scales. There's alpha diversity, which takes into account the richness and evenness that I just explained of individuals within a habitat unit. You have beta diversity, which describes diversity between habitats. And then there's one called gamma diversity, diversity of habitats within a landscape or region. Here you see 3 plots, Site A, Site B, and Site C. And let's define these as 3 different types of habitats. And the shapes within are representative of what a species would be. So for alpha diversity, Site A would have 7, B, 5, C, 7. For beta diversity, you compare 2 sites. You're comparing the 2 habitats together. So A versus B is 8, B versus C is 4, and A versus C is 10. And you're looking for the unique number of species within each. In this situation, B and C are the most similar. And A and C are the most different because they have the least number of species in common. Now, those represent gamma diversity. In this situation, you have 3 habitats, and the total species among all 3 habitats is 12. Now, coral reefs, they're known for their diversity. They're in fact called the rainforests of the sea. And tropical reefs, shallow water tropical reefs, essentially are between 30 south and 30 north degrees latitude. Now in this picture, all the red dots are coral reefs presently, and anything in blue, those are actually the reefs of the U.S. Pacific, or the U.S. territory. So what are within your U.S. jurisdiction? Now, interestingly, these coral reefs comprise of less than 1.1% of the ocean floor. And yet they have 1/3 to 1/2 of the marine biodiversity. Now, just as a little comparison, when I said they're similar to the rainforest, rainforests used to have 14% of land mass coverage. Now it's down to about 6%. They had a lot more percentage of land. Here, it's at 0.1%. Now, an interesting thing about tropical Pacific marine biodiversity is there's this unbelievable gradient of species diversity that has the highest diversity in what's called the Coral Triangle, and that's between Australia and Southeast Asia, and it radiates outward in a decline in terms of richness. And we know this because fish species, there's more than 2,500 in the Indonesia area in the Coral Triangle, but there's only about 1,200 species of fish in Hawaii. And for corals, there's nearly an 8-fold increase of species in the Coral Triangle, with 605 species of corals compared to the 80 species here in Hawaii. Now, there are several factors of why this region would be considered more diverse. In part, it has a lot more area and habitat that was derived from whether it be plate tectonics or changes over time in sea level rise that has created habitats that enabled species to diversify. But we're not talking about fish and coral today, even though they're the dominant things you would see in any sort of image of a coral reef, such as this. Most people see the corals and the fish in the background, but they don't realize that there are a lot of other organisms that reside within this structure. And that's the Cryptofauna. Here is just an example of where you would find Cryptofauna within a reef scale that you would see, such as this image. They're usually found hidden in voids and crevices and blend in our camouflage. So they're concealed. If you break down the word Cryptofauna, it essentially is concealed organisms. And I just threw up these two images here because I thought it was fascinating. They are both shrimps, but the one, it looks like a algae leaf, and the other one just kind of looks like some fuzz. And there are so many organisms that reside within this community. You've got polychaetes, chitons, cowries, nudibranchs, sea snails, flatworms, crabs, clams, snapping shrimp, squat lobsters, you've got abalone, you've got porcelain crabs, hermit crabs, and mantis shrimps. They actually contain the most phyla on a coral reef. So what do I mean by phyla? Now, I don't know about you, but when I was a kid, when we were learning our biology, they taught us a little saying to try to remember taxonomic classification. For me, it was kings play chess on fiberglass stools. Now, what that represents essentially is kingdom, phylum, class, order, family, genus, species. So in this situation, kingdom is the animal kingdom, and phylum is a specific phylum for a group of organisms. And then how they are morphologically and also now genetically related determines on where they fall within each one of these groups. It's essentially like a Russian doll. And so the kingdom's the big one, and then you slowly peel it off until you get to species. Now, phylum is just the singularity of phyla. So phylum Chordata, that's for fish, anything that has a backbone, and humans. And then also there's a Cryptofauna that actually exists within this phylum. It's called the tunicates. And they exist within this phylum because when they're in their embryonic stage, they have a notochord, which is the predecessor to our spinal cord, but they lose it when they become adults. Another phyla, to give an example, is Cnidaria, which are the corals. Now, there are also cryptic organisms within Cnidaria, such as hydroids. In fact, there are so many phyla that reside within this Cryptofauna community, and most people don't realize, but fish and coral represent just 1% of the total biodiversity on a coral reef. The Cryptofauna community collectively has the greatest biomass and abundance on a reef. I just showed one example from a study that was done in the Great Barrier Reef, where he was essentially counting crustaceans, which are like little small insect-looking bugs. They're the amphipods and the tanaids, for anyone who wants a more scientific word for them. And he found nearly 340,000 individuals per meter square on the Great Barrier Reef. That's a lot. Now, to kind of represent what I'm talking about in terms of abundance, this is an image of a coral reef in Moorea. And that green bar is essentially 1 cubic foot. And this photographer, David Lichwager, decided that he was going to do an experiment and essentially count all the organisms within that 1 cubic foot and photograph them individually. This is what he came up with. All of these organisms were within that 1 cubic foot. Now, as you'll see some fish in here. Now, what happened, he decided if there's any sort of fish that swam through that cubic box, he would photograph it. So that's why there are fish there. Now, let's remove the fish. This is the rest of the community. Now, I actually did do the counting. There's 34 fish and 342 Cryptofauna organisms from that 1 cubic foot. They're also extremely important for fish diet. 40 to 70% of the fish diet comes from this community, and people know this because they did gut contents. So if they caught the fish, they'd open up their stomachs and they would look inside to see what's present. And that's how we know they're feeding a lot on the crabs, like this example of the crab in the mouth and the polychaetes, etc. Finally, they're also really important nutrient recyclers. They filter water, they decompose organic matter, they graze the algae. They have a lot of trophic levels present within this community that help sustain a coral reef to thrive. So how do you study this important community? Most people do study coral reefs by doing visual transects. So they lay a transect down and they count the corals, count the fish, or they get pulled behind a boat to do their visual surveys. Clearly, you cannot do that with this community. Now, some people have done live coral head surveys. Here's a representative of someone with their flashlight looking inside a Pocilopora head, and she's looking for all sorts of Cryptofauna that are commensally with this Pocilopora head, but they're counting only stuff greater than 5 millimeters. Other researchers have essentially removed dead coral heads from the reef, and then they break them open with a hammer and chisel and pull every single organism off of the coral head and enumerate them and identify them to try to understand the community. And this is for the motile organism, so anything that moves around. Other people have put settlement plates out to look at sessile, or what you would call the stationary Cryptofauna community. And this would include tunicates and bryozoans and sponges. And they'd place these plates out and then look at the percent cover and the identification of the organisms on plates. Now, there was also destructive sampling. Luckily, I don't have an image of this, but in the past, when you just didn't know what was going on, some people would just completely remove the coral and sift through the coral, because you have different organisms living on live and dead corals. They remove a lot of rubble, they remove chunks of reef to try to assess them. Well, now we have a new method within the last 8 to 10 years called the autonomous reef monitoring structures, otherwise known as ARMS, and I'll be referring to them as ARMS from this point forward. So what are they exactly? Well, they're basically what I like to call reef hotels. They're composed of gray type 1 PVC that are stacked in alternating open and semi-closed layers attached to a base plate. And they're deployed on a reef we deploy them for up to 3 years at a time to allow organisms to settle and recruit to them. So why would we choose this method? Well, it's not destructive. We deploy them on the reef, we leave them there, we encapsulate them, and we bring them back. So we're not taking anything from the reef, we're just taking whatever settles upon them. It's a standardized method. So what this means is that it enables you to compare across whatever gradient of interest you may have. And the methods previously that I described, it's really hard to standardize. So you could say, well, I want to still look at dead coral heads at these different habitats, but the shape of the coral head, the dead coral head, the different species of the dead coral head, how long has it been dead, that's going to influence what has recruited to the dead coral head to look at Cryptofauna. And because it mimics the structural complexity of a reef, you get both the sessile and the motile organisms, so the stationary and the moving organisms that you can look at. Now, this is a movie that really shows exemplarily what the arms units are and how they're deployed and recovered and processed. So you can see here, we're putting the plates on one after the other. And then we take them in a boat. And this was a part of what was called the BioCode project. And it was a 5-year project to essentially census both land and sea as many organisms as possible off of the island of Maria. So they used arms as one of their tools and methods to try to detect and find organisms to study. So you get onto the reef, you pound in these stakes, and then once they're pounded, you kind of just leave them be and go away. Then you come back a couple years later, you can see lots of stuff has recruited to it. It's no longer a nice, clean plate. You put a crate with a mesh lining within it in order to encapsulate everything to avoid anything from escaping. And then you bring it to the surface. Now, once you transport it back to the processing area, you remove the plates, each one, plate by plate, and you take an image of them. And the image basically captures all the sessile organisms as a good reference for later from the molecular techniques. And they're beautiful. They're really like art forms. I always thought they'd be lovely to just put a resin on and then hang on the wall somehow. So once you've taken the images, all the water that was used to remove the plates is sieved into 3 different sieves. This one was the 2-millimeter sieve. Now these are all the motile organisms that you've collected. Now it's the fun process, but also challenging and time-consuming, is essentially you are now putting all of these organisms in their individual cups based on their species presence. Then you go back to the plates, and because there are so many organisms that we do not know or do not have names, we start to integrate genetic tools, something called barcoding, which I will talk about more after the movie. So you take samples of different organisms for genetics later to help you identify. Then you basically scrape all those plates clean. And the reasoning behind this is that there's also a component, then it's a relatively new technique called metabarcoding. So if DNA barcoding is one individual, metabarcoding is multiple. So you create what we like to say a smoothie of all of these plates together, and you subsample that to extract its DNA. So just gonna stop the movie now, because that's basically it. So you have a lot of components from these arms units. You have the photographs, you've got the 2-millimeter sorting, and you've got the DNA work, the metabarcoding. But for the purpose of this talk, I'm just going to focus on the 2-millimeter group, which was identified through morphological just general taxonomics, and then also included some DNA barcoding. But it's still ongoing, as you'll see, for identifying some of these organisms. So I just want to quickly explain what DNA barcoding is. Essentially, let's say you have an organism such as this crab, and you want to find out what it is. Well, you're going to take some of its tissue from, let's say, its leg, and you're going to extract the DNA. So the DNA are the building blocks of our entire being. And the DNA has a code that enables you to essentially identify what you're looking at. When you extract the DNA, after you do that, you need to make lots and lots and lots and lots and lots of copies of the DNA. And you need lots of copies in order to get this sequence. And this is the code I had just mentioned. This is a unique code that's directly to that crab. You get one sequence for one individual. And once you have that sequence, you essentially get a species fingerprint. Or it's also called an operational taxonomic unit, which is a proxy for species, and it's called an OTU. And once you have that DNA and the identification, preferably you get the organism identified by a taxonomic expert. You pair and match it with the DNA, and then you put it into basically a genetic library that stores all these DNA sequences from a number of organisms so that people can access them. So why do we DNA barcode? Here are two images of lionfish. Now, can you tell the difference between them? Are they the same? Are they different? Does anyone know? What do you think?
One is kind of like brownish,
and
the other like more like white, and the other one is like white and more like lighter.
Yeah, I mean, it looks like there's some color difference, doesn't it? But there's also, unfortunately, I was trying to find a better image of the one on the left. The one on the right has a good flash on it, where the one on the left doesn't, right? But they actually are different species, but they're to an expert, they can't tell the difference. Now, because there is some coloration difference, here's a series of 12 images of corals. Some of them look like they've got to be different. Some of them look like maybe they're the same. Essentially, each row is a different species of coral from the genus Pocillopora. And basically what you're seeing here is that some species have what's called plasticity. So they have different features, or their forms are different even though they're from the same species. So it's very hard to identify in the field. Here's another example. So here you've got different colorations. So you may think maybe it's a different species, but they're actually all the same. So this is in part why we want to DNA barcode some of these organisms, because they're very hard to determine what they are. So now that all the background is done, I want to basically tell you where we're putting these arms out. Now, I work for, as I mentioned, NOAA Fisheries, and we are essentially assessed to monitor the coral reefs of the U.S. Pacific Islands, which are shown up here on the world map. We go out on these large boats for 3 months at a time, and we look at the reefs for fish and coral, and we do different climate stuff, such as measuring temperature and looking at water chemistry, etc. But we haven't done anything looking at this crypto community. Very little has been done on them across great spatial gradients, and their distribution is not well known. So we deployed them. We deployed a bunch of units across these different regions that we survey through our assessment cruises. And then we also partnered up with other agencies to have a couple locations in the Coral Triangle. So from this point forward, the colors that you see that represent these different regions are going to stay the same throughout each of the graphs. Now, we had a total of 164 units deployed. Out of those 160, we counted over 30,000 Cryptofauna organisms. We ID'd 493 species that are unique across, basically unique species. And we've reached about 50% of the species identifications only. Now, I want you to understand the magnitude of this. 164 arms units with 493 species and 30,000 counts. That was an equivalent in terms of space with 164 units of 69 cubic feet. That's essentially 3 freezers. That's it. And we're still finding and discovering new things. I mean, that's astronomical. This community is just huge. And it's very difficult to examine as well. Okay, just some graphs to show you some of the information. Now, on the x-axis, the bottom axis, that's the unit abundance. And on the y-axis is the richness per arms unit. Now, the colors are all coded by the regions, and their labels there with the parentheses and the numbers represents how many units were deployed within each region. So as we would expect, which was good, when you have more organisms, you're going to get higher diversity. You're just going to have a higher probability that you're going to see new species. Now, if we look at the regional examination, now here all the numbers of the total organisms counts, that is, are within each of the box plots. And then the parentheses below the regions are the units. And this is essentially taking in consideration this variation in the number of units deployed at each region to try to see what the diversity is across regions. Now, it is significant that it's most likely driven by wake, which had really low diversity. And when you do a pairwise examination across regions, you see that wake is significantly different than the coral triangle in Hawaii, but the rest of them, there's no significant difference between regions. Now, this is what's called a species accumulation curve. And you do these to see how much more do you have to sample in order to capture the entire community. So on the bottom is the number of sampling units. And on the y-axis, on the left-hand side, is essentially richness. Hill equals 0 is a richness factor. Now, this is also extrapolated and interpolated. The reason why you do this is because of the differences in the units. You want to essentially normalize and level them out across the different regions because there are differences. But what this shows us is that when you reach what's called your threshold, the curve is going to flatten out into a line. So you're going to increase as you start looking at each unit. So from 0 to 20, most of them are curved, starting to curve, and it's going to continue to curve until it levels out because you've found all the species. With the exception of wake and possibly the Line Islands, most of them aren't curving, which means there's still a lot of sampling we need to do. Now, this is an interesting plot. This is called a Venn diagram. So what this is doing is taking the species from each one of the regions, and it's looking across regions to see which species are the same. So which ones have we found across each region? And what you can see is that very little, which also signifies how much diversity is out there. So to explain, the percentages within each of the colored wings, so to speak, and then the parentheses underneath it. For example, in Hawaii, you see 25.3%, and with the parentheses, it says 125. What that's saying is that 25.3% of the species from the Hawaiian archipelago are unique to the archipelago from these units, and that's 125 of the total species identified. The coral triangle, it's the 21.5% of everything from within the coral triangle. And it was 106 species. And anywhere it crosses the line, that's the percentage of similarity of shared species. So there's very little. And once you add on wake, it's 0.4% shared species across all 6 of these regions. Well, what's that 2 below the 0.4% is indicating that there are 2 species that are shared. And these are those. So we found Conus obscurus and Thalamatoides quadrascens, so a marine snail and a portunate or a swimming crab. Those are the 2 we found from within each region. That's pretty crazy if you think about when you put the exact same habitat across. And what that indicates is that there's so much more out there that we haven't even captured. Now, Everything that I'd shown you previously was focused on the alpha diversity, which I talked about in the beginning of looking at basically richness and or evenness within a particular habitat. Now we're moving on just briefly to beta diversity, and to refresh your memory, what that was looking at richness, or basically diversity across habitats. So what this plot is showing is it's grouping similar units and sites to similar regions, and it's spreading out ones that are less similar to each other. So in the top right-hand corner, you have the Hawaiian archipelago sites. All of those are clustering together, so they're more similar than anywhere else. On the left-hand side, you have the coral triangle, and they're similar to each other, but less so than everywhere else. Now, for the Samoa and the Marianas Archipelago, they're a bit more intermixed, which basically means overall, their numbers and what we've identified are more similar. Now, so all of that has been focused on a numerical way of looking at diversity. But let's look a little bit about that taxonomic classification that I explained previously. So the phyla, again, is the multiple phylum. Phyla is plural. And here in the arms, from all those units, we essentially calculated 9 phyla from this greater than 2 millimeter community. Now, as you see here, the arthropods seem to be the most dominant phyla that we've uncovered, followed by the mollusks. Now, let's break this down a little bit more, because it's hard to see differences across these regions. So if you go down to families, that was about in the mid of that classification scheme I showed you. We have 177 families that we've identified in total. And this breaks down the organisms that within families of greater than 250 organisms. And the reason why you have to do this is it'd be too complicated to see 177 families within each one of these bar graphs. So essentially, I'm only displaying 10 of them, and all 10 of these have greater than 250 organisms within the family, within each region. So from this, what I take away is that with the exception of the coral triangle, hermit crabs, which is Diogenidae, that's the kind of peach colored one, hermit crabs are the most dominant family across these units. Now, what else do you see there are differences. For example, wake has very little gastropods in abundance compared to the rest of the communities. So we can even just kind of break it down a little bit more. And what I'm going to show you next is just species, but these are species that have greater than 150 count within each region. And when you do that, when you break it down to species, now American Samoa and wake do not have high counts of species, of any particular species. In the Hawaiian archipelago, they have a lot of species of hermit crabs. That's what Calcinus, the genus, is right there. That's a hermit crab. So you can see that there are even the species distributions are quite different, which would be expected since we didn't really see a whole lot of overlap. So why are we even bothering doing all this work to look at this community? Well, there's this understanding that high biodiversity equals greater ecosystem health. So you have 2 reefs here in this picture, surrounded by herbivores, and I'm just using this as an example to explain. Now, let's say the reef on the right, you allow fishing. You know, parrotfish are pretty tasty, so then you're losing your parrotfish. You enable the aquarium trade, so bye-bye tangs. And then all of a sudden, let's say there was a urchin disease. Now, it wipes them out in both environments, but the one on the left will probably sustain itself because it had a lot more organisms that can fill in those voids that were removed from the habitat on the right. And this actually happened in the Caribbean. There was overfishing in the Caribbean, and especially around Jamaica. And then there was an urchin die-off, and the reefs around Jamaica were decimated. And reefs where there were still some fish, they were able to maintain the algae from exploding, because algae and coral are constantly competing with each other for space. And herbivores, they essentially, they're the lawnmowers. They're eating the weeds away from the reef to enable corals and other organisms to thrive. And it's really important to think about diversity and the functions of these organisms, because there are a lot of impacts that are happening on our reefs. These are just naming a few. There's poor water quality, and this is caused by nutrient enrichment, pollution, sedimentation. We tend to overfish our coral reefs. As our population increases, we're just getting hungrier. And we also have a problem with invasive species, which are also impacting the reefs. Now, there's also long-term global impacts, such as ocean warming and ocean acidification. Here in Hawaii, we just experienced an extreme bleaching event in which a number of our reefs crumbled from too warm of water. And it's important to try to not have these impacts just completely destroy the reef, because we use them for sustenance. The majority of human population on this earth lives along a coastline and uses the ocean to help sustain itself. They protect us from waves. They're very good at coastal protection. If the reef starts to break down and there's no barrier, we're going to have much more problems on our coastlines with increased water and wave destruction from storms. They're huge for economic vitality and tourism. They bring a lot of money into a number of states and countries supporting tourism, and they also have some major cultural importance. So I hope from this talk that you've gathered that cryptofauna are pretty abundant and diverse, and we actually know very little of their composition and distribution. High biodiversity equals a very healthy ecosystem. And reefs are actually declining at unprecedented rates. And if we really need to conserve them, if we want to continue having that economic, social, and cultural value that they provide. Now, as you can probably imagine, it takes a village to really support this project. Everyone in red is a taxonomist who has helped with the identifications. People in the black have either supported through field work or helping with some of the barcoding. So it's a continual process, and we are learning so much as we continue examining this community. Thank you, and if you have any questions, let me know. Yes.
Why does Wake have this biodiversity problem?
Why does what?
Wake.
You know, I don't know if it's really a problem per se. I think we need to sample more. And then also, I don't know if you noticed when I had shown basically the table of how many species were actually identified. Wake, we only have identified up to 37%. So we're still in the identification process, but their counts are a lot lower than the rest of the units. So if you have less numerical numbers of organisms in your unit, you're going to have less species. And why that's happening, I don't know. We only had 7 units there, and we need to deploy a lot more.
Yeah, I don't hear of anything that suggests that Wake is under attack.
No, I don't think it's necessarily under attack, but if you want to think about Wake, it is extremely isolated in the Pacific. So it's essentially between the Hawaiian archipelago and the Marianas. Now, yes, the Hawaii is also extremely isolated. However, we've got 8 main islands and 14 northwestern islands and a lot more area and habitat for organisms to thrive. Wake is a small little atoll that has very little area and habitat for organisms to exist. Not only is it challenging getting there, but they're competing the hell out of each other to try to, so that's my thought of why that's occurring at Wake, but definitely need to deploy more units. Yes?
How many sea hotels have you laid out?
How many sea hotels have I laid out? Oh God, I'd say over 500. Because I'm also supporting other people in their projects. So I've been doing this for quite a while, done it all over the world. Yes.
How do you determine on your hotels what the spacing is? Why are they designed with that?
Because the spacers we purchased were a 1/2 inch. And they're easily accessible and cheap. Yeah.
I noticed from the movie you're showing, to the pictures you're showing here, there is a top layer from Phyla.
Yes. We don't, so that was an old model. So that was back in like 2008 to 2011. And we used to have that Matala mat to try to capture even more diversity. The problem of trying to keep that Matala mat was that people were processing it differently. Because some people were tearing it apart and getting the organisms out. Some people were soaking it. So when you have a number of groups around the world using the same unit to do their measurements, removing that from it was the best way forward to maintain the standardization.
I just wondered what other, you talked about around the world, so other countries are doing similar?
Oh, I should have thrown a map up here. I'm sure my boss is watching. He's like, Molly, why didn't you throw the map up? So they've been deployed in the Arctic, in the Antarctic. They've been deployed across the 7 regional seas of Europe. They're in Chagos, they're in the Maldives, they're in Madagascar, they're across the Caribbean. I call it the virus because it just keeps popping up, and we're having more and more units pop up, and it's taking us a while to get all this information through the genetics and the identification. But once we get all that, it's going to be an incredible way of looking across habitats, ecosystems, and just.
But is it just NOAA?
No. So I probably should have also put up here the Smithsonian. They have an ARMS web portal. And so we actually have a pretty extensive website too talking about it, but the Smithsonian does as well. And they're kind of turned into the ARMS hub. And so people want units, they can contact them, and then they will ship them out and deliver it to them. Because when you buy everything in bulk, it's a lot cheaper. It's a lot easier. So that's kind of more, it's like for the US, it's NOAA and the Smithsonian, and there's of course some other academic institutions. And then in Australia, it was AIMS, which is the Australian Institute of Marine Science. And mostly it's academic beyond that in Europe and Africa and et cetera, et cetera.
You probably mentioned it and I didn't hear it. What are the plates made of? And is everybody using plates made out of the same material?
The same material. It's a type 1 gray PVC. Mm-hmm. Yes?
Hi, I'm sorry I'm late and you have a probably great intro, but what's your next step with this? And then what was the budget for it and who funded it?
I'll start with the question, just in case nobody heard this from the live audience, was what are the next steps, and then what is the budget, and how did this kind of transcend? So next steps, I didn't even talk about today, and if you enjoyed this, maybe later in the future, I can talk about the meta barcoding aspect. So there's a whole other genetic component that's diving into the diversity of it. We're linking in environmental parameters. So I gave you just an overall view of regional comparisons. But if you were to break things down into sites where you have more specific information, you can look more at potential drivers of the diversity in these different locations. We're also been doing trophic linkages. So I had a a student who spent a number of days, if not weeks to months, literature to try to get diets, and if they exist, because a lot of information doesn't exist on these organisms, diets and what their probable trophic group is, whether it's a detritivore, which eats all the bottom scuds, or a herbivore, eats the algae, etc., so that you can look at diversity in terms of function and see how functional diversity is changing with different environments. So those are all in the works in the process, just too much to discuss in one talk. Now, in terms of funded, it was originally actually a part of the Census of Marine Life. So if none of you have heard of the Census of Marine Life, it was something that was founded by the Sloan Foundation from 2000 to 2010. And they gave millions of dollars to essentially assess what is the diversity in our oceans. Now, this project had 17 nodes, and a node essentially represented a different ecosystem. So seagrass, pelagics, Arctic, mangroves, and coral reefs was one of them. And so here was this project that was being funded, and you're trying to understand the diversity of a coral reef. We didn't need to target fish and coral anymore. We needed to target this community. So collectively, people who were working a part of that node, the arms were developed out of that. And so then we had money through that project for a number of years, and NOAA at least has been piggytailing on the already expensive costs it takes to do our monitoring, that jumping over and deploying the units doesn't take that much since we're already out there, because it's so costly to get out there. And then the genetics and stuff, I've been funding through grants. Yes?
How many species did they find in smoothies? And was it what kind of organisms?
Oh, the smoothies. So the smoothies is it's a whole other ballpark, and in part it's a whole other ballpark because we're not getting as many species identification. When I explained how when you do a DNA extraction of a single organism and you put that information into a genetic library, you use those genetic libraries to match up to your smoothie extractions. And if there's no books in the library on the Cryptofauna or DNA, then it's hard to get species level. But you can still get that operational taxonomic unit that I explained. So in terms of OTUs, which is the operational taxonomic unit, from those 165 units, I've had close to essentially 250,000 OTUs. Now that's, across a lot of units, right? When you break it down into individual locations, it can range anywhere from 4,000 to 10,000 at a site. Now, if you break it down to things that are actually identities, you're coming down to 100 or so. So there's a lot of work that's in the process on what would be called the bioinformatics. It's a whole nother field of where you're taking these sequences and you're trying to informatically understand how species are related based on the sequences are to each other of known organisms. So that classification tree that I had originally said of the kingdoms, phylum, class, order, family, species, genus, species. Essentially, when you can structure something out of that of known representatives, and then you can essentially what I would like to call plinko, like the Price is Right, and you have a sequence, and you're like, okay, I've got these 5 phyla. What's it more similar to? Oh, arthropod. What's it more similar to in the class? Oh, Crustacea, what's it more similar? And then it can't go further. But it's super complicated, but maybe in several months from now, I can do another one, specifically that.
Going along with that, now you're doing, did plants go into this?
Oh.
Somebody studying plants?
It picks up the plants, the animals, the protists. So you get multicellular and unicellular organisms. You can get the bacteria, all sorts of stuff, all based on the gene or the portion of DNA that you're targeting.
One more.
Uh-huh.
You mentioned that you don't always set them down or leave them down for the same period of time, 1 year, 2 years, 3 years. Do you have any way of knowing if that's going to change your results? You
So-
can leave them down different periods in the same place?
Haven't, everything I presented to you today is 3 years. So
Knowing 3 years is best?
well, you know, that's a really good question, and probably a pilot study should have been done, but we were constrained based on our ship, right? So we go to each one of these regions every 3 years. So
There you go.
there you have it, you know, but no, for sure. But the thing with our logic behind this is that, yes, there is this initial successional state that will happen. So when things are going to colonize an arms unit, initially it's whoever gets there first. Then they're going to start competing. And then over time, whoever wins the competition is probably going to be there by 3 years.
Did you leave any arms out there to be collected, for a 6-year study?
Yes, actually, so our final basically for this project, but our next round mark. So we're skipping the next cycle of 3 years and going to the 6-year mark. And then at that point, we'll reassess whether or not there's continued push to try to do this kind of work, or if we don't receive the money from the, yeah. So it's basically, we have a 6-year mark scheduled.
Is everybody using half-inch spacing?
Yes. Yes, in terms of the actual unit itself, everything is the same. Otherwise, there's no point in comparing across locations.
So do you plan to stick around for that next 6 years, or are you what do you aspire to?
You know, I've been with NOAA now since 2001, so, you know, I'll probably still be here, but I'm assuming I will be here and end up recovering them and then going from there.